Hey everyone, so today we're going to be going over heme and porphyrias. So before we start, I want you to take a look at the learning objectives that your professor gave you. These are really important and they really just show you where to be focusing on, where you should be focusing on while you're going through your lecture. So after you've taken a look, I'm going to be explaining how I'm going to go about this. So for heme biosynthesis, before we can go into porphyrias and what they are, we need to understand how heme is formed. And in order for us to tick off all these points in a way that can make you also understand what's going on, I'm going to be going over the heme biosynthesis pathway and explaining each step. And next to each point that I need you to focus on, or next to each point that covers one of these learning objectives, I'm going to be putting a pink star next to it, just so you can follow along your learning outcomes. All right, so before we start with the heme biosynthesis pathway, there's two things that I want to bring your attention to, which is these 2 points here. So first of all, we need to know where this is all taking place. Where is the heme biosynthesis pathway taking place? The reason we need to know this is because you're required to know which enzymes are mitochondrial and which enzymes are cytoplasmic. So how does this happen? For heme biosynthesis first begins in the mitochondria, then moves to the cytoplasm, and then it goes back to the mitochondria. So it goes mitochondria, cytoplasm, mitochondria. And this is going to make sense once we visualize it better when we go through the actual pathway. The second point I want to bring your attention to is inhibition or regulation. So it's very important to know that heme inhibits its own production. So when we start, so this is our first step, when we start with making heme, and our end result is obviously going to be heme. When we have a lot of heme, our body is going to be like, oh, okay, so we have enough of this. So heme is going to go ahead and inhibit its own biosynthesis. And at which level does it do that? It does this at the first step. So the second thing that you need to know before we start is that heme inhibits its own production at the first step, making the first step a self-limiting step. So far we've covered 2 points. So I'm going to put a star next to this and a star next to this. And I really want you to focus on the enzymes that are going to be coming up, because we didn't actually go into the mitochondrial versus cytoplasmic enzymes yet. All right, so let's begin with the actual pathway. The first step, we have our starting reactants, which are glycine and succinyl-CoA. I'm going to put another star next to this, because this is another thing that you are required to know according to your learning objectives. Your starting reactants, they are glycine and succinyl-CoA. So like we mentioned before, this first begins in the mitochondria. So glycine and succinyl-CoA come together and they form delta aminolevinic acid with the help of two things. So there's two things that should be present, and they are this enzyme over here, the delta ALA synthase enzyme, and vitamin B6 or pyridoxine, along with pyridoxal phosphate, which is an enzyme related to vitamin B6. All right, so already we covered the first mitochondrial enzyme, which is delta ALA synthase. We covered the starting reactants. We covered the fact that heme inhibits its own production at the first step, meaning the first step is a self-limiting step. And yeah, so all of this will also be covered later on, but I just want to go over it really quickly just to give some context. A deficiency in vitamin B6 will lead to problems in the first step of heme synthesis. This is common sense. If we don't have one of the cofactors that are required for this step to take place, then it's not going to take place, and we're going to end up with a type of anemia called sideroblastic anemia, which is a hypochromic microcytic anemia. And I'm going to be explaining later on exactly why and how that happens. This step is regulated by heme and glucose. Again, heme and glucose. More importantly, obviously, heme inhibits its own production, meaning this step is a self-limiting step. Okay, moving on to the second step. So again, we're still in the mitochondria here, meaning this enzyme is also a mitochondrial enzyme. I'm just going to put a star next to this. All right. So we got delta aminolevinate from our first step. 2 of the delta aminolevunic acid with the presence or under the presence of this enzyme will give us porphobilinogen. And this porphobilinogen is called a pyrrole, and it's our first precursor pyrrole. So what does this actually mean? So a pyrrole over here is a ring-shaped molecule with 4 carbon atoms, 1 nitrogen atom arranged in a 5-membered ring. And it's a crucial component of heme that's important for oxygen transport. So I'm going to be going over what a pyrrole is and its importance, and what a precursor pyrrole is into more detail later on. Again, it's going to make more sense with context that we're going to be covering in a bit, which is why I'm going to be explaining this a bit later on. Okay, moving on to the third step. We're still in the mitochondria, meaning our uroporphyrinogen 1 synthase enzyme is a mitochondrial enzyme. So we've got our porphobilinogen. We've got 4 of them. So we need 4 of them under the presence of this enzyme to make hydroxymethylbilane. Again, hydroxymethylbilane is a porphobilinogen, and it's a linear tetrapyrrole, meaning we got 4 pyrroles and its structure is linear. So this is actually here just to clear up any confusion, because in your lecture slides there is more than one enzyme name written, and when I was doing my own research for preparing for this mentoring session, I came across these 2 different names at first, not this one. But from what I understand from how your lectures are phrased, I think your professor wants you to know that this is the main enzyme. But also, do keep in mind that porphobilinogen deaminase and hydroxymethylbilane synthase are also enzymes that could be mentioned. So PBG deaminase and hydroxymethyl synthase. Okay, moving on to step 4. So we've gotten our hydroxymethylbilane. So from here, there's 2 possible things that could happen. This hydroxymethylbilane, this linear tetrapyrrole, could either undergo spontaneous cyclization, or it could undergo a reaction under the presence of this enzyme and become uroporphyrinogen 3. And this is actually what we need to happen if we want to get to heme. So if it were to undergo spontaneous cyclization, it would result in the production of uroporphyrinogen 1. But since we're aiming for heme, we're going to be focusing on this pathway. So under this enzyme, which is the uroporphyrinogen 3 synthase. So now we've officially moved to the cytoplasm, meaning that this enzyme is a cytoplasmic enzyme. Okay, so uroporphyrinogen 3 synthase is going to give us uroporphyrinogen 3. And here is just a note of, just to know like the difference between uroporphyrinogen 1 and 3. The main difference is the arrangements of their substituents. One is symmetric, one is asymmetric. Okay, moving on. So step 5, we're still in the cytoplasm, meaning that this is another cytoplasmic enzyme. So we've gotten our uroporphyrinogen 3. Under the presence of uroporphyrinogen decarboxylase, we're going to get coproporphyrinogen 3. Coproporphyrinogen 3. So once we get coproporphyrinogen 3, we're officially back to the mitochondria, meaning that this is a mitochondrial enzyme. and this is a mitochondrial enzyme. And our last step also happens in the mitochondria. So the only cytoplasmic enzymes are the uroporphyrinogen 3 synthase, uroporphyrinogen decarboxylase. Yeah, okay. So we're here, and we've gone back to the mitochondria with coproporphyrinogen 3. So coproporphyrinogen oxidase, our mitochondrial enzyme, is going to give us protoporphyrinogen 3. Protoporphyrin 3, under the presence of this enzyme, is going to give us protoporphyrin 3, protoporphyrinogen oxidase. So these are the enzymes that I need you to focus on, and that you're going to be required to know that are mitochondrial enzymes. A note that I want to make right now before moving on is that all of this will be summarized really nicely in a document that I'm going to be going over once we're done, just so you can revise on your own time everything that we've covered. All right, so our last step, we're finally gotten to heme. So last thing, we got protoporphyrin 3 from protoporphyrinogen 3. Now protoporphyrin 3 has two conditions that have to be met. You just need iron and ferrochelatase to make heme, and ferrochelatase is a mitochondrial enzyme. And that's it. That's it for the heme biosynthesis pathway. Again, let me just go really quickly over the things that you're required to know. The mitochondrial enzymes, these two, this mitochondrial enzyme, just know that they are mitochondrial enzymes. What they produce over here, this is a cytoplasmic enzyme. This is a cytoplasmic enzyme. This is a mitochondrial enzyme. However, it was not mentioned in your slides, but you still need to know it just in case. This is a mitochondrial enzyme, and this is a mitochondrial enzyme. All right. So if we go back to our learning objectives, identifying the reacting molecules at the start of heme biosynthesis. So we've done this. This is glycine and succinyl-CoA. Done. Identification of the intermediates in the order they form in heme biosynthesis. Although we did not do it the traditional way, which is the listing way. I walked you through the heme biosynthesis pathway. We did technically go through them. I will also go through this point again and take it and give you the intermediates in order, just so it sticks in your mind better. But we technically did cover this point. Identification of the enzymes in the order they react and specification of their intracellular locations, meaning mitochondrial or cytoplasmic. We did this. All right. Explanation of how the rate of hepatic heme biosynthesis is regulated. We did this. Heme. Description of the photodynamic properties of porphyrin. We're going to be going over these 2 points. And yeah, we're basically done. We're basically halfway there. Also, another thing, hepatic heme biosynthesis. I'll go over this right now since it makes the most sense. So, all right. In our hepatocytes and in our bone marrow, erythroid precursors, which are just stem cells that are destined to become red blood cells, and hepatocytes are liver cells. We have this enzyme, the first enzyme we discussed, if you remember. Heme is produced in the liver and in the bone marrow. That's something that you should know. So let me just write it real quick here. Heme is produced in the liver and bone marrow. Meaning that we need this enzyme to be in the liver, in the hepatocytes, and in the bone marrow erythroid precursors, the bone marrow. Now there's different types. the ones in the liver are called ALA synthase or ALAS1. The ones in the bone marrow are called ALAS2. That's it. Here it's specified, they're talking about hepatic, it's the same. Heme regulates its own production both in the liver and in the bone marrow. Alright, moving on. So it makes the most sense to start talking about different porphyrias right now. So that's what I'm going to do. Okay, so let's start with the first enzyme. Let's say we've got over here, this first step. If we have a problem with this enzyme, then this step obviously cannot take place. Either it's deficient, its production is impaired. Let's say you have a vitamin B6 deficiency, because again, this is another condition that should be met. There's two conditions that should be met here. This enzyme and this vitamin. If this vitamin is not there in sufficient amounts, or if this enzyme is not there in sufficient amounts, then this step is not going to be taking place and we're going to end up with anemia. Now, why microcytic hypochromic anemia? So let's break down what that word actually means. Microcytic, micro meaning small cells, small cells, hypo meaning low, chromic meaning color, anemia. Heme is what gives our red blood cells its red color. Just like chlorophyll gives their green color. So if we are stopping at the very first step, if the condition of vitamin B6 or the condition of this enzyme is not being met, then we can't produce heme. If we don't have enough heme, then we can't have enough of the coloring. This is why a deficiency in the delta ALA synthase enzyme is going to result in microcytic hypochromic anemia. Sideroblastic is a type of that anemia. Okay, moving on. So the most common porphyrias, we have three that were mentioned. We have Porphyria cutanea tarda, variegate porphyria, and acute intermittent porphyria. So let's start with the first one, cutanea tarda. This is related or associated with a deficiency in this enzyme, our uroporphyrinogen decarboxylase enzyme. So let's go to that step. So if this enzyme is deficient over here, we're going to end up with Porphyria cutanea tarda. And this is associated with increased urinary porphyrin 1. Okay? This is another thing that you're required to know. You're required to know the clinical presentations of the porphyrias, which enzymes they're related to, and of course their names. So I'm going to go over here. So Porphyria cutanea tarda associated with a deficiency in uroporphyrinogen decarboxylase. Uroporphyrinogen decarboxylase. And presents as increased urinary porphyrin 1. This is our first porphyria covered. Alright, moving on. Our next most common porphyria is variegate porphyria. So variegate porphyria is associated with a deficiency in the protoporphyrinogen oxidase enzyme. So let's go to that step over here. Protoporphyrinogen oxidase is a mitochondrial enzyme, and its deficiency will result in variegate porphyria. Again, which enzyme? The protoporphyrinogen oxidase. All right. So you need to know the name, which enzyme it's related to, and the clinical presentations. What are the clinical presentations of variegate porphyria? Increased urinary ALA, increased urinary PBG, increased urinary coproporphyrin 3, and increased fecal protoporphyrin 9. Moving on to the last one, acute intermittent porphyria. This is associated with a deficiency in the uroporphyrinogen 1 synthase, which is a mitochondrial enzyme over here. So if this enzyme is deficient, we're going to be having acute intermittent porphyria. Okay, so what are the consequences of this? How is this going to be presenting? Accumulation of ALA and PPG in tissues and fluids. So if you have a deficiency in this enzyme, you're going to be able to detect an accumulation of ALA and PPG. So kind of like the opposite of variegate porphyria, where you have increased urinary ALA and PPG. Here you have increased accumulation of these two in your body tissues and fluids. And if you have this, then you can assume that your patient has acute intermittent porphyria. Again, here you just need to know the name of the porphyria, the enzyme it's associated with, and clinical presentation. This is another learning objective that we just covered. Description of the enzyme defects that cause porphyrias and the clinical presentations of porphyrias. We're done. Okay. Now I'm going to be explaining the different structures of porphyrins versus porphyrinogens. So what's a porphyrin? It's a tetrapyrrole ring. So tetrapyrrole. So we went over what a pyrrole ring is. It's just a 5-membered ring. So 1, 2, 3, 4, 5. 4 of these are carbon. 1 of these is nitrogen. And there's 4 of these rings. 4 of these rings will make up a porphyrin. Another important thing that you need to note about porphyrin is their double bonds that they have between these pyrroles. And why is this important? It's because these double bonds are what give them their photodynamic properties. These double bonds are what give them their ability to absorb UV or just absorb light. And it's what give them their distinct color. Now, porphyrinogens are essentially the same structure. They have similar structures to porphyrins, but they have additional hydrogen atoms at specific positions. Now, you're not required to know exactly what these specific positions are. You just need to know that the difference between porphyrins and porphyrinogens is that porphyrinogens have these extra hydrogen So the main difference is the hydrogenation of the pyrrole ring. So again, without these hydrogen atoms, this is a porphyrin and it has a distinct color and it's able to absorb light because of its double bonds. However, if we take the same structure and we hydrogenize it, we add a hydrogen atom at specific sites, it's going to become a porphyrinogen and it's going to be colorless. And that's the main difference structurally and the photodynamic properties of porphyrins. So we just did this. and this one. Okay, we're left with our last point that we need to cover. Explain the significance of the laboratory in diagnosis and prognosis of porphyrias. Before moving on, I actually want to go over this page really quickly. It just summarizes what I just explained. So porphyrins, due to the double bonds between the four chiral rings, is able to exhibit its photodynamic properties. In the same way that chlorophyll is responsible for the green color of leaves, heme is responsible for the red color of red blood cells. Specifically, it's the porphyrins that give this ability to heme to color the red blood cells. Alright, so laboratory diagnosis of porphyria. Alright, so there's two ways we can go about this. We can either test the urine or the feces. We test the urine for uroporphyrins and we test the feces for coproporphyrins. Now, just an important note, uroporphyrins are not only found in the urine and coporphyrins are not only found in feces. This could be found here and this could be found here, but we're sticking to the majority. And the reason I'm mentioning this is because this point could come in a which of the following is false question, where your professor might try and trick you and ask you which of the following is false and put one of them as coporphyrins are only found in the feces. That is false because coporphyrins can also be found in the urine, but mostly found in the feces, which is why we test these respectively. Alright, so if we go about testing the urine for uroporphyrins, or if you choose to go about testing the feces for coproporphyrins, there's three methods you could follow. Either the simple screening test, spectrophotometric scanning, or fluorometry. Simple screening test, spectrophotometric scanning, and fluorometry. Again, another thing that you are required to know for exam. This is another point in your learning objectives. If you choose to go with simple screening tests, then this would be the positive result or an indication of a positive result for porphyrins. If you choose to go with spectrophotometric scanning, then a presence of a Sori band would indicate the presence of porphyrins. And if you choose to go with fluorometry, again, it was not explained exactly what or how these tests are performed. So I'm guessing you just have to know the names. But if you're going to go with fluorometry, then these would be the positive results or the indicators of a positive result for the presence of porphyrins. That's really it. So simple screening test, spectrophotometric scanning, and fluorometry, testing the urine for uroporphyrins and testing the feces for coproporphyrins. Just like that, we covered all the learning objectives. So now I'm going to be going over the summarized document that I've prepared, just so we can wrap it all together really nicely and make sure that we have all our points straight. So this is the summarized document. Over here, I just have the learning objectives listed off with the simplified answers. So I'm just going to be going over them really quickly. We already covered this. Knowing the starting reacting molecules of heme biosynthesis, they are glycine and succinyl-CoA. And here I just have some extra details that I did already go through and are helpful for extra context. The second learning objective is identifying the intermediates in the order they form in heme biosynthesis. So over here, when we're referring to intermediates of heme biosynthesis, we're referring to the porphyrinogens that are involved in the pathway, in addition to protoporphyrin 3. Although it is not a porphyrinogen, protoporphyrin 3 is still considered an intermediate. Over here, I have them listed off in order. And below this, I have the steps of heme biosynthesis with all the enzymes underlined and the actual intermediates in bold. Moving on, here it's asking us to identify the enzymes in the order they act and the specifications of their intracellular locations. Again, just asking us to know the mitochondrial versus cytoplasmic enzymes. Over here, I have 6 enzymes. The ones that are underlined are the ones that were mentioned in your lectures, and the ones that are not underlined were not mentioned, but they're important to know. Over here, I have the cytoplasmic enzymes, which are the uroporphyrinogen synthase 3 and uroporphyrinogen decarboxylase. And again, here I have the same exact steps that we went through, except the mitochondrial enzymes are sectioned here, and the cytoplasmic enzymes are sectioned here. Over here we have description of the structural differences within the different porphyrinogens and between porphyrins and porphyrinogens. So we went over this. We said they have similar structures, with the only difference being that porphyrinogens have hydrogenized pyrrole rings, and they are colorless, and the double bonds of the porphyrins and their lack of hydrogen atoms is what give them their ability to absorb light and their photodynamic properties. And these are actually the 2 points that were mentioned that you're required to know with respect to structural differences within the different types of porphyrinogens. So porphobilinogen is a linear molecule with 4 pyrrole rings, and hydroxymethylbutane is also a tetrapyrrole, in contrast to these 3. So that's porphobilinogen and hydroxymethylbutane, which are linear tetrapyrroles. Over here we have explanation of how the rate of hepatic heme biosynthesis is regulated. Again, ALAS1 in the liver. Heme and glucose negatively inhibit the first step of biosynthesis, making the first step a self-limiting or a rate-limited step. We discussed this. Again, description of the photodynamic properties of porphyrin, description of the enzyme defects that cause porphyrias, and the clinical presentations. So again, I explained this, but this is just a simplified or summarized version of what I went through. So we have deficiency in this enzyme, what it causes, and its clinical presentations, deficiency in this enzyme, this one, and this one. So there's 4 total enzymes, and 3 porphyrias that you need to know, because one of them causes the hypochromic microcytic anemia that we discussed, and 3 of them cause the most common porphyrias that again we discussed earlier. Okay, lastly we have explanation of the significance of the lab results in diagnosis and prognosis of porphyrias. I mentioned that we test urine and feces. We test the urine for uroporphyrins and we test feces for coproporphyrins. And these are the 3 methods which we use: simple screening, spectrophotometric scanning, and fluorometry. And that's it for heme and porphyrias. If you have any questions, please feel free to dip into the Discord server that we have and type in your questions, and I'll be happy to answer them. And good luck.